In the last unit, we looked at cell division, and we looked at it from a number of perspectives. First with mitosis and the importance of cell division in order to, again, allow for growth and repair, as well as the creation of new cells. And then later with meiosis and how that form of cell division permitted organisms to carry out sexual reproduction. Now, for many years, scientists wondered what was the mechanism that was allowing cells to be able to know how to carry out many of these complex processes. And Darwin suggested there was an internal mechanism. Gregor Mendel later showed that there was an internal mechanism, but we still, going into the 20th century, we really didn't know what molecule inside the cell permitted cells to do all of the complex jobs that they were known to carry out. Okay. Now, in the 20th century, scientists began to take a closer look at cells and the molecules that were inside of cells, trying to narrow down, which one of those molecules was the one that was primarily responsible for driving, cellular instructions. Oswald Avery in 1943 was one of the first ones to show what molecule actually was doing it. And in his experiments, he showed that this was accomplished by a molecule that scientists had kind of overlooked in the past, DNA. And what Avery did was he was able to take different organic molecules, from lipids to polysaccharides, polysaccharides being, starches and somewhat complex sugars, as well as proteins and well as nucleic acids. And what he was able to do was he was able to manipulate bacteria so that by inserting these various organic molecules into the bacteria, he was able to transform the bacteria and give it traits from another bacteria, an original one. And what he learned from his experiments was the only molecule that was able to do this transformation was DNA from the original one. So when he inserted lipids, nothing happened. Polysaccharides, nothing happened. Proteins, nothing happened. But only DNA carried it out. So this was the first inkling we had that DNA made a difference. Now, his experiment, began this process. In 1952, A.D. Hershey and Martha Chase carried out experiments that were able to basically confirm Avery's initial idea that DNA was the molecule to focus upon, and their work provided, again, additional proof, scientists always looking for verification, that DNA was the primary mechanism for driving the instructions that cells utilize in order to carry out the complex jobs that they do. And so their experiment, a decade after Avery's, was able to really show scientists that DNA was the molecule that was the primary director of what happens inside cells. In their experiment, they labeled DNA and the protein, from a virus, and they gave them radioactive markers. And what they did was they found that when the virus was able to create new viruses, they found that basically the part of the virus that was in the copied viruses that were created inside the cell, inside the bacterial cell, the part that was copied was the DNA and not the protein. So even though both were given the markers, what ended up getting copied into new cells was the DNA. And so this basically narrowed down scientists' view of the importance, of what molecule provides this essential information. And so we're going to spend this lesson looking at DNA. We've begun to do that with the film we've started, Race of the Double Helix, and we'll come back to Watson and Crick a little bit at the end of this slideshow. But, understanding DNA became the central dogma of what scientists invested lots of time and energy in the last half of the 20th century. So the first thing we need to understand is, what is the job of DNA? Its function is to provide all of the information that a cell needs in order to function and carry out every job that is necessary for cell survival. At a different level, what it basically provides is the information that a cell needs to make every single protein that the cell will require in order to build the enzymes, to build the molecules that are necessary for cellular homeostasis, for cellular functioning. Now, at an even more smaller level, we now know and we now use the term gene to describe a specific part, a specific length on a DNA molecule that contains the information, the very specific information for how to build just one protein. So every time you hear the word gene, you need to think instructions for one protein. And so if we say we have 20,000 to 30,000 genes, that means on our DNA, on human DNA that is, that means we have the information for building 20 to 30,000 different types of proteins inside our cells. Okay? And so we recognize that DNA is the set of instructions, and those instructions are written in this DNA molecule. Now, scientists, again, began to investigate this DNA molecule, and one of the things they wanted to understand first was, how could the instructions be written in the molecule itself? And that led them to look for, how was the structure of DNA, put together? How was it built? So you can see this molecule here, and we know now that the molecule itself is coiled into this double helical structure, and every cell has this DNA, and we now know that every single DNA molecule is made up of individual components that are referred to as nucleotides. Now, each nucleotide inside a cell is made up of the same basic components. Each nucleotide is made up of a deoxyribose sugar, a phosphate group, and a nitrogen base. Now, as we'll also talk about, there are different nitrogen bases, but we'll come back to that in just a little bit. But each nucleotide is made up of these these three components, and a DNA molecule is made up of millions and millions of these nucleotides bonded together in a sequence. And again, a sequence of nucleotides eventually leads to what is known as a gene, and multiple genes are found on each of these DNA structures. And so as we begin to break down the DNA, we begin with looking at these individual nucleotides. Now, as scientists have realized and learned, these nucleotides are bonded together in a sequential order. One of the things that scientists needed to look at is, how are they composed? Now, scientists now know that that each nucleotide is put together with the sugar and the phosphate, okay, on the outside of the DNA molecule, and the nitrogen bases built into the inside, okay? And so when scientists look at this DNA molecule, what they realize is that, okay, the sugar, deoxyribose, and the phosphate provide the backbone to basically hold the molecule together. And so there are two of these backbones, okay, each one running along the outside of the DNA molecule, and as we now know, the nitrogen bases are aligned along the inside, okay? And so it becomes known as the double helix because of these twin sugar phosphate backbones. When scientists, again, that was one of the big questions that scientists were investigating as they were looking for the structure of DNA. You know, how was it put together? As we know now, Watson and Crick figured out that it was this, okay, double helical structure with the double backbone of sugar and phosphate on the inside, and the nitrogen bases eventually on the inside. Now, one thing that became relatively clear to scientists as they began to study the DNA was that it was the backbone that just provided the means to hold the molecule together. Since there was this regularity to it, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on, it could not provide the cellular instructions because there was really no variation there to provide something different. So that led scientists, particularly those like Watson and Crick, to look at those nitrogen bases, okay? And what scientists kind of begun to wonder was, you know, when they looked at these nitrogen bases, there were really ending up being some questions about what and how, you know, could these provide, okay, the information. Now, a scientist by the name of Erwin Chargall eventually, okay, in the 40s, provided one of the important clues about the value of these nitrogen bases when he discovered that the amount of adenine and the amount of thymine, okay, were equal to each other in every DNA molecule that he studied, and the same could be said for the amount of cytosine and the amount of guanine. Now, these became known, okay, this information that Chargall discovered became known as Chargall's rules. And eventually what scientists realized was that the reason for these equalities was that in a DNA molecule, adenine chemically always ended up getting bonded to thymine, and cytosine, due to its chemical nature, always ended up getting bonded to guanine. And so adenine with thymine, cytosine with guanine. And so therefore, in every DNA molecule, on one side of the structure, attached to one part of the backbone would be adenine. The only thing that could attach to it would be thymine. If thymine was on one side, adenine had to be on the other. And the same thing with cytosine and guanine. And so this led to several understandings, okay? And what scientists realized was that the nitrogen bases will always match up like this. And so depending what scientists realized, particularly Watson and Crick, was that the pattern of these nitrogen bases must somehow provide a code for telling the cell how to build a protein. And so, and what they also realized with that was that, you know, the unique way that these molecules bonded also gave them a big clue as to how the DNA was able to copy itself when new cells were made. Now, a number of other scientists played a role in putting the whole big puzzle together. One of those scientists, as we'll see as we've begun to see in the film, was Rosalind Franklin. She discovered that DNA was twisted into the shape of a double helix. Now, she was well known and well respected for her work in X-ray crystallography, which is basically the ability to use X-rays to take pictures of organic molecules in order to determine their structure by the pattern that's created as the X-rays bounce off the molecules. And it was her X pattern, okay, that was taken of the DNA that she was working with that first provided scientists with the knowledge that the DNA was built into this double-stranded, double helical shape. Right? Now, Franklin deserves a lot of credit for kind of laying the groundwork for how the general structure was put together. Okay? And so when we talk about a double helix, it was that knowledge came through the groundbreaking work that she accomplished in her lab, okay, in England, okay, but she did not put the whole picture together in terms of the role of these nitrogen bases. So when we look at, again, a DNA diagram, a structure of the DNA molecule, what we can see is, again, this double helical shape, and we can see how the two halves wrap around each other. Question became, at some point, why does it wrap like this? It turns out that the ability to wrap into this double helical shape and then continue to twist itself into these shapes allows the DNA to actually be coiled and supercoiled into the nucleus, into the inside of the nucleus, so that it can fit there. But it was Rosalind Franklin who first gave us the final understanding of this. Now, eventually, okay, the two scientists who kind of put together the whole big picture of how, you know, the sugar, phosphate, nitrogen bases are organized, what's the importance of these nitrogen bases, okay, what the entire structure looks like, okay, how that structure can possibly indicate or provide instructions for the cell, as well as how it could possibly provide a mechanism for the cell to copy the DNA. That was Watson and Crick. They were the first scientists who really put together, okay, this information. They were synthesizers, and they used the work of, okay, Chargall, Franklin, and others to truly build the first correct model of DNA. And you can see them in this picture pointing to it and looking at it. And they published their work that explained the structure as well as how cells can copy it in 1953. And this accomplishment, okay, on this small paper published in Nature was so significant that both of them, okay, won the Nobel Prize, okay, in science, in biology, in 1962. And as a result, we now say, we now know the structure of DNA, how it's copied, and we also know how proteins are made, which we will look at in this unit.